Everyone. We would be starting with the talk in more 5 minutes. And in the meantime, you can introduce yourself in the chat box. And hi, Julia. Good morning.
Muted. Hello, everyone. Thank you very much for joining. We're just waiting for 5 more minutes for everyone to join. So just requesting your patience for a bit more. Thank you, everyone, for joining again. So we will just wait for 1 more minute to make sure that everyone who wanted to be here is online. So let me just begin by introducing everyone to Dr.Taranenko. She's a researcher at Tauber Bioinformatics Research Center at Haifa University in Israel. And I'm really happy to welcome you, Lisa, also here. Thank you for joining. So the workshop is going to be hands-on. So whenever you have a question, it's best if you're already registered on the platform and you can follow along on your computer. Lisa will be sharing some data that you can also run on the platform. If you don't have an account on the platform, you are welcome to register on the platform and get an account. So Nalika, can you just write in the chat, please, the email that everyone, thank you very much. So you can, in the chat, you can see the email you can write to if you have some issues with an account to the platform. If you have any questions, you can write in the chat and we will try to answer them as we go along. I'm sure that Lisa will not have, might not have a lot of paying attention both to the slides and to the chat, but we will help her along as much as possible. So I think we're good. Thank you, Lisa.
Thank you very much for having me. Yeah, as Julia already mentioned, we will be going through a metagenomics section hands-on with preferably trying it ourselves. But if not, you can use this presentation and materials later on to try it on some data that you want. Okay, then let me start sharing my screen. Okay, can you see my screen?
Yes, it's visible.
Okay, thank you. So yeah, I will start with general overview of the pipeline that we have for our metagenomics section. So we have actually subsections in there, and when we start analyzing our data, which is raw reads in FASTQ format, we have to, of course, pre-process them. Then after pre-processing, when we have cleaner reads, we can create abundance table. So just know how many of the specific ID we can find in every sample. Then we assign taxonomic group to each ID, and we can finally calculate core metrics, some alpha and beta diversity there, and visualize everything and finally look at the results. Okay, I have the chat window open as well, so if you have any questions, feel free to write and we can stop there. And first thing I wanted to speak about was where to find the data to run this analysis yourself. There are two approaches that you can, well, there are two types of data that you can use this platform with. And one is when you have data yourself, you sequence them yourself, or this is the newest data that you got from your colleagues. Or actually you can surf the internet and find some data that might be interesting for you, and you can analyze that on the online. And I would like to start with this bio project. I will add the link to the chat. Wait. So you are on the same page as I am. So this is a project about Engelmann syndrome in rats and in mouse. And how we find it, we just Google a bunch of things in NCBI, try to find the data set that you're interested in. But from here, we actually have to move to SRA Run Analyzer to get the metadata that we need for input. And here I'll give you steps how to find this file, because it can be tricky. So first we can look what kind of data this is. We see the registration date, it's really recent one. We see who did these data sets, and we can see how many samples we have there, 75 samples, SRA experiments. So we click on that and we move on to another information about this data set. This is specific one of the runs there, and a bit more information about what actually was sequenced. The instrument that was sequenced, that was used, type of this analysis, the strategy, the amplicon. There's also it's also possible to, for example, sequence whole genome sequences. Metagenomic, then we have this one, layout, which is very meaningful for us. It's single end or paired end reads, and we will need it for our analysis. And also construction protocol, which is also very meaningful. We have to pay attention what primers were used for amplification and what region was amplified in our data set. Okay, let's just remember that we will need this data and click on the run. And we will end up on the SRA website where, again, we can view all this information again, but we need all our 75 samples in one page. So let's click on SRA study, and we'll have another information about what this project is about. And here you can see 75 runs and the total memory usage that was used and everything. We need this. So after we click here, we finally are on the SRA Run Selector webpage. We again have all the information about our experiment. And here you can see the metadata. So basically, this is information for each run, each sample, where which organism it was taken from, what is the genotype, if it's wild type, or if it's the one that is interesting for us, and the host, well, a lot of columns there. What we need right now is to download the metadata file. And I hope you're on the same page as I am. Please let me know if I'm moving too fast. We can slow down. So after you download the metadata file, it's normally in TXT file, it's comma separated or it's tab separated. But if you open it in Excel or in numbers, for example, it will look like a table. It will look like something like this. But let me move to another window for a bit. Yeah. In fact, it will look something like this. So it's pretty huge. They have all the information here that we might need. So it's really your concern if you need all this, or if you can just take parts of it, especially if they're the same, take parts of them and just remove this information altogether, because it will be pretty hard to navigate later on. But please pay attention to some entries that are interesting. For example, the host common name or the host scientific name, doesn't matter. If, for example, your research is more interested in rats than in mouse, you can only pick this rows and discard mouse, or vice versa. Doesn't really matter. So I encourage you to look through your metadata and to filter out columns and rows that are not interesting to you, because they might become troublesome later on. In my case, I was interested only in mouse, and I was only mainly interested in a few columns, especially the genotype and, well, since I filtered not this, but okay. So this is my final table. You can see that I only have mouse here already. Is it? Yep, so only mouse here already. And the most interesting for me is genotype. And I left the rest just in case. And so with this file, we can proceed. You have to export it. So it will be tab delimited or comma delimited, but it should be a TXT file. It cannot be Excel file or something like that. Okay, back to the slides. I'm not sure if everything goes well with this file for you. So I will send this file in the chat. It's possible, right? It's possible. Sorry, Julia, can I actually send the file? I don't think I can.
You can send it to me and I'll try to send it while you continue.
Okay.
You know, just send it to me and I'll send it.
Okay, thank you. Yeah. Thank you for your help. Okay, so let's assume you have this file. Oh, so Zoran, you successfully did everything. Okay, so let's move on from here. If you have this file ready, and hopefully Julia will send it if you don't have this file ready, so you can try it out anyways. We will have to move on to the TBio platform. Send, you already have a link, but I will just send it. Wait, no, if this link works. And in the main menu, we go at the very end. The link that was sent by Sonalika, and you have to pick Metagenomics Chime 2. And the first thing that we have to do is to specify what type of input we have. So there are two inputs. I already told you that you can have manual input when you have all the files, the FASTQ files on your computer, and you will be uploading them directly. Or you can use NCBI upload when you only have the metadata, the file that we just looked at together, and the system will automatically download all the FASTQ files according to the first row, which has the IDs, which is pretty handy. Also, things that you have to remember is, what is the strategy? Is it single end or paired end analysis? And in case you have the manual upload, there is also a possibility to choose if it is multiplexed or demultiplexed data. Multiplexed and demultiplexed means that you have one or several samples in one set of files. If it's one sample, then just FASTQ files are all right. If there are several samples, then you will have to have a metadata that will help you differentiate between samples inside these files. However, if we're working with NCBI upload, it is always demultiplexed, means each file that will be uploaded, it will be on its own. So we pick NCBI upload, NCBI upload and single end, and clicking sequences. On the sequences, we have to upload our TXT file that Julia just sent the link to in the chat. Please download it if you don't have your file ready yet. So just upload it, click options, and we're moving on to the third tab in the upload system, that is to pick the variable region. So for this, I'd like to step away from the platform a bit and talk about what is actually being sequenced in case of amplicon sequencing. 16S RNA has several variable regions that are along the whole 16S region. And there are already primers that are mostly used for each region, and some regions that are very popular. For example, one of the most popular are V4 or V3, V4, the combination or anything. It is advised to use the same primers as were used for sequencing. Sometimes for sequencing, they change the primers a bit, it's possible. Or for example, they don't exactly use 341 forward primer, but maybe they shift it a bit. That also can slightly affect your results. So it is best to use the same primers as were used for sequencing. But if you don't know what exactly are the primers, like in our case, they just give you numbers, they don't give the sequence, or even they just give you the region. Then on the platform, you can pick the region that that is mentioned in the study. For example, for us, it is written that it's V4 region, so we can just pick V4 region. Or if you know a specific sequence of your primer, here you can pick custom region and just add the sequence for forward primer and reverse primer. Okay, so you are on the platform, you're not sure where to go to the upload. Okay, we're not in rush, so let me share my screen. Okay, can you see it?
Yes, we can see.
Okay, so I'm also on the platform, and here you have to scroll down to the metagenomics section and pick metagenomics CHIME. It will take you on this website, and here's the upload system. Here we can choose. It didn't work. Not sure what is the reason. Okay, maybe someone can help Zoran, because I'm not sure this is the issue of the platform. Okay. Then I'll go back to my slides. So yeah, after we picked the region, in our case, it will be just V4, the easiest way, and it will automatically add the primaries. We hit submit, and we can start building our pipeline. Let's just go step by step, preferably together. So first, we import the data into CHIME. This option doesn't really need any parameters from you. What it does, it just adds all our information into CHIME program. The next is filtering samples. What it does is three options and options. If you are talking about demo, then you don't have to choose a demo. You can just choose Metagenomics CHIME 2. If that helps. So what this job does, it filters our samples to have at least some number of reads in all of them. And if we do not meet these requirements, the samples are filtered out. So we have to have at least 100 reads in our samples to keep them. And the last one in the pre-processing is to have the quality control plots and graphics to understand if actually the trimming that was done is okay with us, or we need to have a stricter trimming there. Okay. Next subsection is pre-processing OTUs, getting the abundance tables for our OTUs versus samples. Here we have two algorithms that are basically denoising algorithm, both data2 and DeBlur. In this case, we can use data2. And next is Silva classification. So when you were picking your variable region, this is where it will be used for Silva classification. The same reads that you entered will be used to extract the same regions from Silva database that already has taxonomical groups assigned to these sequences. And yeah, no more options needed, because you already picked your variable region as before. The next part is when we want to remove or preserve some specific taxonomical groups. For example, we don't want any eukaryota or mammals. We only want to have something up to second level to the phylum. We can choose this. Or actually, you can go through the file, the Silva database, and see how the taxonomic file is arranged. And you can use the specific names of the bacteria if you want to only have them, for example, or you can similarly exclude specific bacteria if that's also your idea. Well, then we do the phylogeny and move on to core metrics. And this one is a tricky thing because here you have to pick a sampling depth. What sampling depth actually does. At the moment, we did not integrate an automated way to determine sample depth. So with the new files, it sometimes takes several runs to understand what is the optimal sample depth in your data. Sampling depth is a number of reads that will be in all our samples in order to normalize all our samples. Because, for example, if in one sample you have 20,000 reads and in another one you have 50,000 reads, the proportion can be different, and we don't want to really work with very different numbers later on, because that will affect our analysis. We wanted to normalize them to have uniform sample size, let's say like that. And for that, we come up with a number that will work best to preserve the most samples, but at the same time, and preserve the most unique features, unique OTUs, but at the same time, not have all the reads, because at some point they're just redundant and they're not giving us any more unique OTUs. So with this number, probably you'll have to play if this data set is new. And alpha rarefaction is actually the tool that helps you understand what is the optimal sampling depth for you. Here we already integrated an automated way to determine the maximum value for alpha rarefaction. And you can then view these results and try to understand what is the optimal sample depth. So after we're done with this subsection, we can move on to visualization subsection. It has alpha diversity, beta diversity visualization, and interactive visualization. So with alpha diversity, all the metrics are calculated at the previous step on the core metrics. And here we're just creating files to visualize all that. We don't have to choose any options here. But for beta diversity visualization, you have to choose which column you're actually interested in. You can change it later on, but the statistics will be calculated for this column, which you can't change later on. So, for example, in my case, I'm really interested in the genotype, so I will be picking the genotype here. And for interactive visualization, I am interested also in the genotype and, for example, the sex. If you're interested only in one column, you can pick it just twice, and it will just have one group and not two groups. And that is it. We're done with our pipeline, and now you will have to wait for the results, and it may take time. So I can give you a link to the pipeline that already worked, so we can look at the results together. Okay, I'm sending the link. And let's wait for a few minutes so you can open this link, and maybe someone can tell me if they can use this link. All right.
Yes, it seems like it's working.
Okay, then I'll start. If something is not clear, just stop me. So our main results will be held in, let's say, two places. Just to download the files as done by the QIIME pipeline. And you also can look at the dashboard, the interactive dashboard. First, I wanted to show the files because just to explain the logic. Each step, each job that we looked at is outputted as a zip folder, and you can download, for example, self-classification folder and look into its files, or for example, pre-processing and look what kind of files are there. Or you can download all the files that have QIIME, all the QIIME files, and see through them what exactly do you need. But it is tricky to look at QIIME files, although we output also TXT files, but still, it's a lot easier to look at the interactive dashboard that you can get to if you click on the external dashboard. I think I'll move there as well. Okay. So this is the pipeline. And I click interactive dashboard. Okay. And we have three main windows. The taxonomy bar plot that shows us the abundance of different bacteria in all our samples. And you can see here the groups that are actually interesting us, the wild type and the group of interest. You can navigate through this, where you can see the sample name and the percentage. You also can choose which taxonomic level you want to look at. let's say film or something else. And we can also check what we want our group as at the bottom of this plot. You can try and download this as a picture. Sorry, I forgot the word. You can download this as a picture or as a HTML file or a table that is behind this graph. So this will give you an idea of what is actually inside the file, the sample, how the abundance of the bacteria and all. Another type of data is beta diversity principal coordinate analysis results that we can see here. And our color is genotype. And my symbol, I chose it to be sex. If you don't want it, you can use, again, genotype. It will only have 2 variables. If you want to have several, you can choose several. It will have 4 now. It will have different symbols for the sex. And you can look through it. And yeah, in this case, we were not very lucky. We don't see any separation here. Well, it happens. And also for alpha diversity, you can also choose which column you want to see your differences in. This is observed OTUs here. And again, although we see that it's not as wide as for our group of interest, there is no significant difference in our sample at this point. But you can learn your quantiles here, the numbers, the median value, and everything. What is this data based on is a table of abundance, the one that was used after the rarefied table, the one that was done after you came up with the sampling depth, and it was unified across the table. And you can also download this table. You can export exactly this one, or you can export your taxonomy data, or just rarefied table, or the metadata, anything you want. And it's also very meaningful to export this one because apart from these, here, the family name, for example, family name is already integrated. Or if you want it to be an order, then you will be having an order. In these tables, you will have to yourself try and merge them together to get this information. This is the dashboard, and I think it's very helpful. But at this moment, we're still working on how to improve our dashboard, and not all the utilities are in there. And I'll go back to my presentation again to show how you can look for, so we looked at this, how you can look for other data that is also given by QIIME. So for example, if you upload, if you download this file that ends with QIIME QC, you will have a file named Dmx sequence 2. Well, 2 is, this is in my case because I have a lot of files like that. You'll just have a file that will end with QZV. And that file you can upload to a website. One moment to send it. Named QIIME View. The idea of this website is to, here, the website. The idea of this website is to visualize all the files that QIIME prepares, because in QIIME there are 2 types of files. It's QZA which have like tables and everything, and QZV that are made to be visualized in this viewer. And for example, the QC part, where we wanted to see the quality of our reads, we upload this file to the viewer, and here's our plot, where we can see that, yeah, well, as we could have expected, we have some quality drop at the end, and we could work on that. Or, for example, if I'm interested in more alpha diversity metrics, not only observed OTUs, but additional metrics, because they're for alpha diversity metrics. We could also download the zip folder with alpha diversity visualization and look through our QZV files. For example, upload I believe this is evenness and metric index. And you can also see the box plots here and the statistics, if this was significant or not. In our case, not significant. So that's it from my side. Maybe if anyone has any questions, we could go through them. And if not, then yeah, I believe this will be available later on, and the presentation as well, so you can try it out yourself. Yeah, happy you liked it.
Thank you very much for very informative talk. And anybody who has questions, you're welcome to write them down. Lisa will have a look. And next time, Lisa will present single-cell sequencing analysis on T-Bioinfo platform, so you are welcome to join. We will post this new Zoom talk on the page, and we'll send everyone a message about it.
I'm sorry, can I take a moment? I see that there is a question if it's possible to use this platform for virus research. Exactly, QIIME is not, it cannot really be used for viruses, because for taxonomy, we use bacterial data set. But I think if you change the taxonomical table to the one that you want, that will have actually viral sequences, then it is possible, but it will require some manual coding.
In addition, I just want to, oh, sorry, do you want to finish?
No, it's fine. No, it's fine.
I also wanted to just let everyone know that there are several sections of the platform dedicated to analysis of virus genomes, not on QIIME, but on and just genomic variants, haplotypes of viruses. So we have at least 2 sections that I can think of that you can use for analysis of virus genomes. Thank you very much for joining again. Thank you.
Thank you for your time. Okay, bye.